Students, welcome to lecture 17 in biopharmaceutics, and today's lecture is from the chapter in vivo in vitro correlation IVIVC, and the topic is factors to consider for IVIVC. So the outline for various topics which will be discussed in this lecture are factors to be considered for IVIVC studies. So the important factor would be BCS, which we will discuss, and simply it means biopharmaceutical classification system. Then internal predictability and external predictability concepts involved with IVIVC studies. So the important factors which can affect IVIVC studies includes the important one is biopharmaceutical classification system, also called the BCS. So BCS is a drug development tool that allows estimation of the contribution of three fundamental factors, including dissolution solubility and intestinal permeability, which governs the rate and extent of drug absorption from solid oral dosage forms. So that's why you can understand that why it is so helpful. You can see that with the help of BCS. Okay, students, I hope that I don't need to go into the detail of BCS because we already discussed in a sufficient detail. So moving on to IVIC, you can see that BCS has particularly it depends upon important parameters which govern bioavailability and dissolution as well. So like solubility and intestinal permeability. So BCS to consider this system is very helpful whenever we are performing IVIVC studies because it gives a lot of information about the nature of the drug and its possible anticipated behavior as far as its bioavailability or resolution is concerned. BCS becomes a basic tool in determining the conditions under which IVIVC are anticipated, as we just discussed that on the basis of knowing the nature of the drug, whether it falls under class 1, class 2, class 3 or class 4 drugs, you can easily anticipate its behavior. Behavior like in terms of resolution or bioavailability. Because if a drug is highly soluble and highly permeable, you can easily anticipate that its bioavailability can easily cross more than 90% and it will be highly bioavailable. And that is the same reason that most of the time FDA gives a waiver for such drugs. Moreover, it's also used as a tool for developing the in vitro dissolution specifications. Bioavailability studies for determination of IVIVC. Um, Bioavailability studies are very important to conduct because if you look at this terminology, it is basically it is the interrelationship of in vivo or correlationship of in vivo and in vitro studies. And even if you are using it for the future reference, still you need to conduct bioavailability studies first to find out the correlation. So a bioavailability study should be conducted to determine the plasma concentration time profile for each of the formulation. And then sufficient number of subjects is important to conduct such studies in order to determine the performance of the formulations. Here what they mean is that smaller number of subjects sometimes are highly subjected to subjective results, or you can say that the results could be not that authentic because of lesser number of subjects. And as you know that bioavailability or the pharmacokinetics of drugs usually vary from one individual to the other individual on the basis of various factors. So a sufficient number of that's the main reason that sufficient number of subjects is extremely important whenever you are considering in vivo and in vitro correlation studies. So determination of predictability of IVIVC. So whenever IVIVC studies are performed, one of the hallmark property of such studies is their ability to predict. To predict what? To predict in vivo performance of a drug product from its in vitro resolution characteristics is maintained over a range of in vitro resolution release rates and manufacturing changes. Now let me explain it. Um, it means that whenever IVIVC studies are performed, which means in vivo in vitro correlation studies, this must be capable to accommodate if any further changes in dosage form are brought about, like the manufacturing changes they are referring to, or for example, any resolution rates changes, in vitro resolution rates, which would definitely come out because of different changes in manufacturing, bringing changes in the process or in the excipients, or for example, if a different apparatus is being used. So this correlation which you already found out, that should be able to accommodate these changes, these range of changes, and still it must be able to predict whatever is going on in terms of in vitro resolution rates and its correlation with in vivo. So whenever such predictabilities are made on the basis of IVIVC studies, so it must be kept in mind that depending on the intended application of an IVIVC, which is definitely to find out the correlation, and in future to go out for only the in vitro resolution tests and avoiding the in vivo uh, studies. So basically, depending on the intended application of an IVIVC and the therapeutic index of the drug, evaluation or prediction error internally or externally may be appropriate. Internally or externally just means that within the body or in vitro, like outside the living body. So therapeutic index of drug is why important. It is important because you are predicting bioavailability, and you want to go for uh, ultimately a stage where you can perform the in vitro studies and just avoiding in vivo studies, which are definitely authentic and reliable studies and which give the actual bioavailability or absorption characteristics of that dosage form. But you want to avoid that in future. That's why you are performing in vitro, in vivo correlation studies. So keeping in mind their intended purpose and also the drug in question, if the drug is having a low therapeutic index, um, that could be a potentially important issue. And looking at the nature of drug, your error for predictions decreases too much, and you have a very limited scope or limited, you can say, spectrum of errors. Your predictions should be very, you can say, accurate, and there are lesser chances of errors, because if you are making the errors, you are taking the wrong chances. For example, you are dealing with the digoxin, and you know that digoxin is a drug with a very narrow therapeutic index, and it could cause very fatal adverse effects or toxic effects. So in that situation, you are on the basis of your wrong predictions, you could be taking the risk of life of the patient. So that's why I think you can understand that whenever IVIVC studies are performed, along with the BCS, and other important parameter is the therapeutic index of drug, which also gives a very important information about drug's nature, how much safe is this, or how much chances you can take in the case of such a drug. External predictability evaluation is not necessary unless the drug is a narrow therapeutic index or if the internal predictability criteria are not met. Thus, prediction error internally is inconclusive. That's the same thing we discussed already. So internal and external predictabilities play a very important role whenever IVIVC studies are being performed, and specifically when various levels are being dealt with. All IVIVC should be studied regarding internal predictability. One recommended approach involves the use of the IVIVC model to predict each formulation's plasma concentration profile from each respective formulation dissolution data. That is, it is very simple approach that IVIVC model is developed, and each formulation's plasma concentration profile is then predicted from each respective formulation's dissolution data. And as you know that already the IV study has been performed, so means in vivo study has been performed. So in such a way you get very sure about this correlation, and this is what you call internal predictability, because it is based upon um, you can see the in vivo or the bioavailability or drug dissolution within the living body. So it is sort of internal predictability. External predictability, as far as is concerned, it is not necessarily limited to only the products which are under question or under study, but it could be what it simply means that it could be externalized to a range of products of subsequent lots of drug products. So that is what you call external predictability. So most important when using an IVIVC as a surrogate for bioequivalence, as we already discussed, that the ultimate goal of IVIVC studies is to become able or to attain a point when you are able to um, assess various dosage forms, um, profiles of their in vivo behavior on the basis of already conducted in vivo in vitro correlation studies. Now there might be changes coming in the products, there could be manufacturing changes, changes within the dosage form based on the excipients, or changes in the manufacturing procedures. But whatever is the change, but changes are coming, but still you are able to predict on the basis of IVIV studies which you conducted. That is what you call external predictability and measurement, and to how much extent your study or IVIVC study can be externalized to a range of products. That's, that is what you call external predictability. So how it could be tested out, and this involves using an IVIVC to predict the in vivo performance for a formulation with known bioavailability that was not used in developing the IVIVC model. So here comes that when the IVIVC model was being developed on the basis of dissolution rates, externally in vitro dissolution rates and its in vivo behavior, their correlations were set up. There was a range of formulations or products. Now those could be 3 or 4. Now what is done in order to establish the external predictability in a much better way, and other formulation with a non-bioavailability is selected, and then in vivo performance for a formulation with non-bioavailability is predicted on the basis of this model, IVIVC, or you can say that the in vitro resolution rates which you determined in your IVIVC study, those are used for a very different product which was not used in the study at all, but which is having a known bioavailability. So now the in vitro dissolution rates of your study will be compared with that one, and it would be giving you an estimate that how closely your in vivo in vitro correlation model is able to detect uh, on the basis of your previous studies this product which has a non-bioavailability, and how precisely you are able to predict on the basis of your IVIVC model. So this is a cross confirmation too. You can see in a way of your IVIVC model or your conducted study, that if you could pick it up and you are able to apply, or it is applicable to a number of range of products having the same you can say active ingredient or the same formulation, let's say, but with different excipients or different procedure in which has been involved. So this is what you call external predictability, and this approach is usually adopted in order to assess that how reliable your external predictability approach is. That IVIVC includes in vivo relevance to in vitro resolution specifications and can serve as and support the biowaivers, as we discussed in the start, that the basic purpose of IVIVC studies is to serve as a reference for not to perform the bioavailability studies again and again and support the biowaivers. Furthermore, IVIVC can also allow setting and validating of more meaningful resolution methods and specifications. It can also assist in quality control for certain scale-up and post-approval changes. So the last one means that sometimes a product is developed, but manufacturing company wants to bring some changes in its excipients or its manufacturing style. So it is what you call scale up and post approval changes. So that could be easily brought about in the presence of IVIVC model. This could happen easily.